All right, welcome to Let's Get to the Marks. We're looking at the AQA Biology GCSE paper 2 from 2021. It's a 1 hour and 45-minute exam. I can't read, never mind. Let's go. Make sure you pause the video and have a go, and then go through the walkthrough and answer the questions. You can do that question by question. All right, let's get into this. The nucleus of a cell contains DNA. Name the structures inside the cell nucleus that contain the DNA. Now, the answer can be the chromosomes or the chromatid. Now, what you can see here, I've drawn a chromosome, and here's your wiggly little double helix of DNA inside the chromosome. And chromosomes, when they replicate, form X shapes. And at that point, when they form X shapes, we call each of the individual copies of each chromosome chromatids. So that's why that's an alternative answer. All right, figure one shows part of a DNA molecule. Here you've got DNA molecule. Name the part of the DNA molecule labeled X. So what is X? Well, X is this kind of pentagon, five-sided shape. And X represents a sugar, specifically a pentose sugar. So you can get the mark for saying, oh, it's a sugar. You can get the mark for saying it's pentose, or you can get the mark for calling it deoxyribose. You're not allowed to call it ribose. That's only found in RNA, and this is a DNA molecule. So it's either a pentose, a sugar, or a deoxyribose. Right, let's look at the next question. 1.2. Name the part of the DNA molecule labeled X. We've done that. 1.3. Whoopsie. What type of substance are labeled A, C, G, and T in the figure 1? Well, what is A, C, G, and T? They're known as bases. If you're A level or you think you're advanced, you call them nitrogenous bases. Now, the names of the specific bases in DNA are adenine, cytosine, guanine, and thymine. So you could give any one of those names and you would get the mark. It's only a one marker, but any of these five possible answers will get you the mark. Let's zing. Right, here we got figure 2. We can see you've got a DNA molecule kind of unzipping and something's going on inside there. 1.4. Four of the substances you named in question 1.3 are not located in part Y of figure 2. Label each of these substances with the correct letter A, C, G, or T. Use information from the other parts of the figure to help you. So I'm drawing arrows here. These are the 4 blank areas. They're saying, what should the letters be that go in each of the blank areas? So pause the video and have a go. You might have heard of the term complementary base pairing. What's happening here? New bases are lining up as this DNA is replicating. And C will always join with G. I always say Cecil and Gary. And T always joins with A, Terry and Harry. Yeah, that's just my way, but you can think of any way to remember those. So G and C, A and T. Okay. And that's worth 1 mark. Now let's zing down to 1.5. What is happening to the DNA in part Z? So we've got some words here. We've got differentiation, evolution, fertilization, or replication. Well, what do these words mean? Well, differentiation is what stem cells do when they specialize to become other cells. They differentiate. And then you've got to think, what is fertilization? Well, that's when a sperm and egg join together. What's evolution? That's the changing of species over time. The answer is replication. The DNA is copying itself by lining up 3 nucleotide bases by complementary base pairing to make 2 daughter strands of DNA from the original. We're actually, the DNA, a new DNA strand is being made, and we're using the original as a template there. Okay, right, 1.6. A gene is a length of DNA. What type of substance does a gene code for? There's a couple of possible answers here. So genes code for proteins, specific proteins. You can say polypeptides, which is just a name for a long chain of amino acids. What you can't say, by the way, is that genes code for amino acids. A protein or a polypeptide is made up of many amino acids. Bases like adenine, thymine, guanine in the triplet code of bases, that is what codes for each amino acid. You're going to get a whole load of bases in a gene, so a whole load of amino acids. So you can't say it codes for an amino acid. Right, let's look at 1.7. Most human body cells contain 6 times 10 to the minus 12 grams of DNA. So that's the mass of DNA found in the nucleus. What mass of DNA would a sperm cell contain? Right, well, a sperm cell, you might remember, is haploid, contains half the number of chromosomes, so half the DNA. And therefore, we would expect a sperm cell to weigh half as much as the normal human body cell. Well, its chromosomes should weigh half as much. So essentially, we're told above that 6 times 10 to the minus 12 is the weight of the chromosomes in a normal human cell. So just divide that by 2. Make sure you stick that in the calculator, and the answer is 3 times 10 to the minus 12. What is the name of the type of cell division that produces sperm cells? And we've got 4 possible answers here. We've got binary fission, differentiation, meiosis, and mitosis. Well, binary fission, triple scientists, you know this. This is how bacteria reproduce. So that's not the idea when a sperm cell's made. Differentiation is how stem cells, those embryonic stem cells, specialize or differentiate to become cells with an adaptation for a specific job. Mitosis, that's part of asexual reproduction in some plants and organisms, and it's for growth and repair. So meiosis is your answer. And I always think the E in meiosis stands for eggs and the S for sperm. So that's my way of remembering that meiosis is for making eggs and sperm or gametes in other organisms. Right, let's move on to what have we got going on here? A student investigated the effect of temperature on the decay of milk. Figure 3 shows the apparatus used. We have a thermometer, we have a pH sensor, and we have fresh milk and a pH meter connected to a computer. This is the method used. Set up the apparatus as shown in figure 3 with the milk at 20 degrees. Record the pH over 5 days using the computer. 3. Repeat with another batch of fresh milk at 25 degrees. So you can see up here, we're changing the temperature. That is our IV, independent variable. And then we're measuring the pH. What you measure is your dependent variable. How could the student keep the milk at a constant temperature for 5 days? Well, you use a water bath. Whether you're doing biology, physics, or chemistry, a water bath will control your temperature. 2.2, give one variable the student should have kept constant. Don't refer to the temperature. That's the independent variable. That's what they're changing. So they're changing the temperature between the different batches. So we can see, what about the amount of milk? Well, you can write amount of milk, you can write volume of milk, or you can write type of milk. You might think, what do you mean by type of milk? Well, milk from different companies or different cows, or goat milk, cow milk. You could have all different types of milk. So keep the milk the same, as it will have the same amount of proteins and chemicals inside it. Okay. Oh, of course, you've got semi-skimmed, full fat milk. You've got all different types. So you want to keep the milk, the type of milk the same, or the volume, or the amount. Lovely jubbly. Let's move on. Ooh, we got a graph question. Scroll up and read this graph. Table 1 shows the students' results for the milk at 20 degrees. We've got the time in days, 0 to 5 here, and then we've got the pH that was measured on each day. You can see the pH is going down. Complete figure 4. You should label the x-axis. That will get you 1 mark. Use a suitable scale for the x-axis. I'll talk about that in a minute. Plot the data from table 1 and draw a line of best fit. So what we want to do here, I can't really squeeze the writing in here, so I'm just going to write on the side. You would put this underneath. This is time in days. So the unit is days, and you need to put that. You can't just put time. So time in days, that would go below. Now we also, that's going to score us a mark. Now we need to use a suitable scale. What they mean by a suitable scale is it's got to take up over half the graph paper. So if I just choose 1 centimeter, that won't be a suitable scale. So a suitable scale is like this: 0, 1, 2, 3, 4, 5, takes up over half the graph paper, and that gets you your second of the 4 marks. Look how easy that is. So yeah. Now the third one is probably the hardest, a mark to gain, and that is to plot the data. You are allowed to mess up one of your results here and get it half a little square out. Okay, so that's fine. You don't have to have eagle eyes. You just got to get it pretty accurate. So at 0, we go up and we got 6.7. And then at 1, we got 6.7 again. At 2, we have 6.3. So I'm just matching this up to the left-hand side, the y-axis. 3 days we have 5.3, then 4 days we've got 4.8, and 5 we have 4.4. So that is going to get me the mark. Even if my circles are half a square out, you will still be given the mark. Now, draw a line of best fit. Now, do we do a straight line or a curve? Well, this is biology, and when it can fit a nice curved line, we can do a curve, and there we go. So we don't need, that's my curve of best fit, and that will get me 4 marks. Boom. Awesome. Now, for the next part of the question, you need to sketch a line as if you did it at a higher temperature. So let's see how that goes. So the 2 marks are for drawing your new line below the original line and then finishing in the same place. And why is the line below? Well, we have the reaction is happening faster, so the pH is decreasing more quickly. However, you're getting the same amount of product as we haven't changed the amount of milk, so it ends in the same place. All right, let's move on.
Keep moving on, keep moving on, keep moving on, keep moving on.
Okay, thanks for that little music. Let's get into question 3. The human body temperature, question 3, is controlled within very narrow limits. Scientists investigated the effect of drinking ice cold water, oh, that'd be nice, on the internal body temperature and the rate of sweating. This is the method they use. Sit a person inside a room kept at a constant temperature of 25. Measure the person's internal body temperature near their brain. Measure the person's rate of sweating. After 20 minutes, give the person 500 centimeter cubed of ice cold water to drink. Continue to measure the person's internal body temperature and sweating rate for a further 50 minutes. 3.1. Give a reason why the person should not move during the investigation. Well, if you're moving, you are respiring. You are releasing heat. Heat, well, respiration is an exothermic reaction for those chemists out there. So you're going to release heat and you're going to affect your temperature, which could mess up our investigation. As one of the things we're measuring, one of the independent variables is measuring the internal body temperature. So obviously, if you move about, that could be affecting it. All right, let's get into this. So now we've got a graph, figure 5 and figure 6 here, and they show the internal body temperature and the time in minutes at the bottom. And you can see after 20 minutes, the person drank some ice cold water, and about 5 minutes after that, or 10 minutes after that, their internal body temperature dropped. So that's what the first graph shows us. Now it climbs slowly back up, or quickly back up, however you want to read it. And then if we look at the next graph, this shows the rate of sweating, how much they were sweating. So they drank the water at 20 minutes, but they didn't really get hardcore sweating until 30 minutes. So this graph shows us all that data. Let's have a look at the question. And we can see from 3.2, what is the person's normal body temperature? Well, we saw that above. I labeled it on the graph. It was 37.4. Now, the results show that when the ice cold water was drunk, the temperatures near the brain decreased. Explain why the temperature near the brain actually decreased. And this is for 2 marks. So obviously the cold water, when the person drank it, went into their stomach and it absorbed heat from the person's blood. So their blood temperature decreased. Where does the blood go? It travels around the body. So it would have gone to the brain, and that was detected as a decrease in the internal temperature near the brain. Now let's look at 3.4. The thermoregulatory center in the brain responds to the decrease in temperature. How does the thermoregulatory center send information to the sweat glands? Well, it sends information via impulses along neurons for your second mark. Let's look at 3.5. The rate of sweating changes between 24 and 36 minutes. Explain how this change helps to maintain the person's normal body temperature. So if we look at the rate of sweating between these times here, how does this rate of sweating help them achieve normal body temperature? Well, what's happened? It's gone down. We can see that between those times, the rate of sweating decreased. Now, zooming in here a little bit, how does this actually help maintain the normal body temperature? Well, if you're not sweating as much, that means there will be less evaporation. The evaporation of sweat decreases your body temp. So if you're not sweating as much, which they're not, That will get you 1 mark, which means less evaporation. That will get you the second mark. And of course, less evaporation means less heat removed from the body, which helps increase your body temperature back towards the normal. Now let's check out 3.6. Let me get rid of all this old answer. During exercise, the skin appears red. What causes the skin to appear red? Blood vessels move closer to the skin, constriction of the blood vessels, decrease in heart rate, or dilation. Well, constriction of blood vessels means getting narrower. Would that make you redder if the blood vessels near your skin got narrower? No. The answer, of course, is dilation of the blood vessels in the skin. And what happens is the lumen, which is the space in the blood vessel, widens, allowing more blood to flow to the skin so you can lose more heat. So the answer is the dilation of blood vessels. And it's an adaptation. When you do exercise, you go red in the face because you dilate the lumen of your blood vessels, allowing a greater blood flow to the skin and a greater heat loss. Okie dokie. Question 4. Decay happens in a compost heap. We got a compost heap with layers of dead plant. Smeared in between are thin layers of soil. So you've got your dead, rotting grass, and then in between each layer of dead, rotting grass and leaves, you have a thin layer of mud or soil. And the question is a 6 marker. Describe how microorganisms in the layers of soil help to recycle the chemicals in the dead plants, and then how the chemicals in the dead plants are used by the living plants. Now, as in all of these 6 mark questions, when there's two sort of parts to the question, you want 3 or 4 points on one and 3 or 4 on the other, and that will guarantee you the 6 marks. You will need a minimum of 2 points on either to get the full 6 marks. So you could have 4 on the first point, 2 on the second, or you could have 2 on the first point and 4 on the second. It's easier to aim for 3 to 4 for each. So microorganisms like bacteria, fungi, decompose the dead plants or decay them. They release enzymes that are things like protease and amylase that humans have in their body. And these digest large molecules like proteins or carbohydrates into smaller soluble molecules. Proteins get digested by protease to amino acids, and starch gets digested by amylase to glucose. Now, what do they use the glucose for? Well, the glucose is used for respiration. That's what the microorganisms do, and they release carbon dioxide into the atmosphere. Okay, so those chemicals are released into the atmosphere. They also release minerals such as nitrates, magnesium, phosphorus, and phosphates into the soil. Now, the second part of the question is asking us. So we've explained how these chemicals like carbon dioxide, nitrates, etc., are recycled. But how is it that the living plants use these chemicals? Well, living plants absorb carbon dioxide from the atmosphere. Remember that was released by the bacteria when they did the decay. They absorb that through their stomata by diffusion, and they use it in photosynthesis to make glucose in their bodies, which they go on to make starch and large molecules. They can make cellulose in their cell wall. They can add it to nitrates to make amino acids and proteins, to build structures within them. And they can add it to magnesium to make chlorophyll, which is another one of the minerals released during decay. Now, plants in their roots use active transport to absorb minerals. Okay, active transport is a process which involves the use of energy from aerobic respiration. And essentially, nitrates are just an example of one of the minerals they can absorb, and they can join those with amino acids to make proteins for growth and repair. And so this would definitely get us 6 marks. Just add in here that magnesium is needed for chlorophyll. You could talk about phosphorus or phosphates used for DNA and things like that. There are lots of different minerals that plants need. Okay, so there we go. Time to move on. And as I say, you'd need 2 or 3 of these on each point. Right, let's move on to the next question. Whoa, pages, pages, pages. Question 5. The growth of daisy plants on a lawn is affected by biotic factors and by abiotic factors. Figure 5.1, Table 2, question 5.1, Table 2, shows 6 factors. Tick one box in each row to say whether the factor is biotic or abiotic. Well, first of all, you need to know what biotic and abiotic means. So, biotic means alive or living factors, and abiotic means non living. So, just go through and put the ticks where there's living things. So, rabbits eating plants and people trampling plants, those are both caused by living things, and the others are all not living things. Now, we've got a bit of a difficult question here. Which involves a bit of maths and a bit of quadrating. So let's get into it. A student estimates the number of daisy plants growing on the lawn. Figure 8 here shows a plan of the lawn. The student places a quadrat at 10 different positions on the lawn. The quadrat measures 50 centimetre times 50 centimetre. The student counts the number of daisy plants in each quadrat. How should the student decide where to place the quadrats? Pause, have a go. All right, let's give a reason for the answer with the answer. Best if I don't type in white because that won't show up. Let me just zing back and rewind. Let's do that in black. So why do they place the quadrats and how do they decide to place them? So they place them randomly. You could shut your eyes and throw them around, or you could draw a grid, number the grid, and use a random number generator on your calculator to do this. Why do we place quadrats randomly? The answer is to avoid bias. So the 2 marks are place the quadrats randomly to avoid bias. 5.3, here comes the difficult one. The mean number of daisy plants is 6 in each quadrat. Calculate the number of daisy plants on the lawn. Give your answer to 3 sig. And it's a 6 marks maths calculation. Okay, so you can pick up lots of marks without getting the full marks, but you've got to show you're working on each step of the way. Let's go. Okay, we need to find the area of this lawn, and let's just put the data down that we needed. It was 3 significant figures the question wanted us to do it to. There were 6 mean daisies in each quadrat, they told us. And we also, above in the stem of the question, when I was reading through, we learned there was 50 times 50 centimeter was the size of the quadrat, 50 times 50. So what we really want you to focus on now is to pause the video and see if you can calculate the total population of the daisies in that triangular field. I'm going to go through the answers. So first of all, I'm converting my quadrat into meters because the size of the field is in meters. So I'm going to do 0.5 times 0.5, and that gives me 0.25 meters squared. So that is my area of one quadrat. Now let's work out the area of the triangle, and I need to do width times height, but because it's not a rectangle or a square, I need to divide that by 2. You should know how to work out the area of a triangle. So I'm going to do the width, which is, or length, however you want to do it. I also need to do the width times the length, and I need to minus that paved area as well, and I need to divide by 2. So let's do it. We got 6 plus 6 plus 4. That gives me a total, what's that, 16 meters. So we've got 16 meters wide, and then the length is 10 meters. So I'm going to do 16 times 10. That gives me 160 meters. I need to divide that by 2 to get 80 meters. And then I need to subtract the size of the paved area. Now, the paved area you can see is 6 meters long, 3 meters high, is 18 meters. So I'm going to do 16 times 10, which is 160. I'm going to divide the 160 by 2 to get 180. Sorry, 160 divided by 2 is 80 meters, and then we're going to take away the 18 meters. So the total area of this triangle is 62 meters squared. So now I know I've got 62 meters squared. If you wrote 80 meters, you've forgotten to subtract the paved area. Okay, now what we need to do is, we know the quadrat is only 0.25 meters. So how many quadrats would fit in 62 meters squared? We'll divide 62 by 0.25. You'll get the total amount of quadrats that will fit in it, and it's 248. Remember that the average number of daisies per quadrat is 6. So there's 6 daisies in each quadrat. We've just found out there's 248 quadrats. So do 248 times 6, and you will get 1,488. daisies, and that's the estimation of the population for 6 marks. But there are all of that working. Each step there is scoring you a mark. So there's plenty of marks to get, even if you mess up one part. And they'll do error carried forward as well. Aha! Look, I only got 5 marks. I forgot 3 sig. I just realized. So three significant figures, we would round this to 1,490. Okay. Okay. Using the mean from this investigation to calculate the number of daisy plants on the lawn might not be accurate. Why is it not accurate to use the mean? Although they placed 10 quadrats over that lawn, the quadrats could have randomly all ended up clumped together in one corner. And that means that the mean might not be representative of the number of daisies spread across the whole field or lawn. We also have to realize that the quadrats were small. They were only 50 by 50 centimeters, which is a 0.25 meter squared quadrat. It would have been better to use a 1 meter squared quadrat because they weren't actually sampling a very big area if you add up the total size of all their quadrats. Right, question 6. Reflex actions are coordinated by the nervous system. What's meant by a reflex action? Well, a reflex action is rapid and it's unconscious, which means you don't have to think about it. You can use the word involuntary because if you're like me, you can't spell unconscious. So it's a rapid, unconscious response to a stimulus. You can say an involuntary, rapid response. A woman's hand accidentally touches a hot object. The woman moves her hand away rapidly. Describe how the woman's nervous system coordinates the reflex action. So, the first part of this rapid reflex is that the receptor cells in the woman's finger touching the hot object will detect the stimulus, which is the hot object. Now, these receptor cells create an impulse, which is a small electrical message or signal that's going to travel along the sensory neuron until it reaches a gap between the two neurons called a synapse. Now, at the synapse, the message goes from being electrical to chemical. The chemical transmitters cross that gap called the synapse by diffusion and bind to receptors on the other side at the next neuron. The next nerve or neuron is known as the relay neuron. That's the one you can see here just running through the vertebrae of the spinal cord. Now, not all reflex arcs have 3 neurons. Some just have a sensory and a motor neuron, but some have this relay neuron going through the spinal cord or brain. And the impulse then travels down the relay neuron and crosses another gap, just like before, called another synapse between the relay and sensory neuron. And then it enters the motor neuron. It travels down the motor neuron to the muscle, which we call an effector. Once the impulse reaches the muscle or effector, it causes the muscle to contract, and the response is for the woman to pull her hand away from the hot object. And just as a side note, sometimes these reflex arcs don't end in an effector called a muscle, they end in the other type of effector known as a gland. The impulse will reach a gland, and the gland will secrete a hormone that goes on to have a wider, less localized reaction, a longer lasting reaction, like the release of adrenaline. But all of these ones where you mechanically pull your arm away from a dangerous stimulants as a response, they tend to result in a muscle contraction. So the effector is a muscle. Please note here that I probably should have mentioned the term that a synapse is a gap between two neurons, as I have implied it in the answer by saying the synapse is between the neurons, but its definition in GCSE is a gap between two neurons. Right, let's move on. 6.3. The endocrine system coordinates many internal functions of the body. Give 3 ways the coordination of the endocrine system is different by the coordination of the nervous system. Right, the endocrine system is this system that releases hormones. It kind of works in tandem with the nervous system a lot of the time, but hormones are a lot slower, and that's one of the points. They're not as fast. Also, hormones travel in the blood. They're soluble chemicals that dissolve in the blood, whereas impulses are electrical signals traveling along neurons. Also, hormones have a widespread effect. For instance, when you get scared, all your hands stand up over your body, whereas the nervous system's effects are localized. You touch a hot object, you whip your hand away. Also, when you touch a hot object and you whip your hand away, it lasts milliseconds, whereas the hormonal response from the endocrine system is a lot longer lasting. For instance, When you get scared and your heart beats fast, it takes several minutes for your heart to return back to its normal beat. Okay, describe how hormones control the menstrual cycle. And that is 6.4. Let's go. So we have follicle stimulating hormone, otherwise known as FSH, and that ripens or matures the eggs in the ovaries. It also stimulates the ovaries to produce estrogen. Estrogen is an ovarian hormone, whereas FSH is produced in the pituitary gland. Estrogen causes the lining of the uterus to thicken. So FSH ripens or matures the eggs in the ovaries. This causes the ovaries to produce estrogen, which develops the lining of the uterus. Interestingly, the estrogen operates by negative feedback and inhibits any more FSH being released. If you think this is sensible, why would you mature or ripen more eggs? It would be a waste. If you're now getting ready for a possible pregnancy, you don't want any more eggs to ripen. So the estrogen inhibits the action of any more FSH and reduces it. The estrogen also stimulates the release of luteinizing hormone from the pituitary gland, travels through the blood to the ovaries, and causes ovulation, which is the release of an egg. That happens around the 14th day. The LH, the luteinizing hormone, also stimulates the release of progesterone. Progesterone maintains the lining of the uterus, and progesterone also inhibits the release of any more FSH or LH. So, if you are pregnant, FSH and LH will not be produced. And that means you won't ripen any more eggs or release any more eggs, which would be a waste if you are pregnant. Because if you're pregnant, your progesterone levels remain high and your estrogen levels, and they inhibit the FSH and LH. And that's really how the menstrual cycle is controlled. And you would easily get your 5 marks there. Okay, 7. A food web contains several food chains. Figure 9 shows a food web. 7.1. The animals in Figure 9 get their energy by eating other organisms. Describe how the algae get energy. Pause the video, have a go. Okay, you can see the algae circled here. They get their energy from the sun by photosynthesis. They actually contain chlorophyll, which absorbs sunlight, and they use that to make glucose and to all the things that organisms like to do with glucose, like respiration and building proteins and things like that. Now, for the next question, we need to name one of the primary consumers. Primary consumers are the things that eat the producer. And so, our primary consumers that are in Figure 9, you've got three things that eat the producer. We've got krill. So I'm going with krill. There's two other things up there that do. And name one producer. Well, that will be the algae. They're the thing that actually makes the food. So I'm going to go with the algae. 7.4, you basically got to point out which food chain above has five organisms, including the human. So which of these has five organisms, including the human? And you can go algae to krill to squid to mackerel to human, and you'll find. Awesome. Just speeding through at this point. There's the five organisms in the food chain. Let's move on. Zing, zing, zing. Where are we? Where are we? 7.5. Figure 9 shows the mackerel eat the krill and the squid. The biomass of mackerel. Sorry about this. Let me just write in the 5 steps in that food chain. So we've got algae, krill, we got the squid, then we got the mackerel, then we got the human. The biomass of the mackerel is much less than the combined biomass of the krill and squid. One reason for this is the mackerel cannot digest all the parts of the krill and squid. Give two other reasons why the biomass of the mackerel is less. Well, what you have to remember with the mackerel is although it's eating the squid and eating the krill, it doesn't get to add all of their energy and mass onto its body. And that's because there are a number of things it's doing. It's doing excretion, so it's getting rid of feces, urine, it's losing mass like that. It's respiring, so it's losing mass via carbon dioxide. It does movement. In order to hunt the krill and the squid, it has to move, and that's going to use up energy. So when it eats them, not all of the energy from them goes on to its body to make new cells. It has to move and hunt and all of that kind of business. Also, it's got to keep warm, and by doing that, it does respiration to release heat and keep warm. Right, we're going to move on to the next question. So Figure 11 shows how the biomass has been changing between 1950 and 2010 in the North Sea, and this is of herring. So we've got the biomass of herring on the y-axis on the left, and we've got the year down below, 1950 to 2010. Calculate the percentage decrease in biomass of herring between 1960 and 1977. Give your answer to a whole number. 4 marks, boom, boom, boom. You might want to pause the video and have a go. I'm going to get on here. So each of these tiny baby squares is worth 2 years. So we want to be somewhere in the middle of the fourth square for 1977. Draw that across there. That's zero point 0.1 is the biomass of the herring in 1977. So that's not good. 0.1 million tons, especially when if you look in 1960, there were 2.3 million tons of herring in the North Sea, down to zero point 0.1 million tons. Well, that's not so good. What is that percentage decrease? So what you're going to do here, you're going to do 2.3, take away 0.1, you're going to get 2.2. So we're going to want to do 2.2, which is the change in the biomass, divided by the original biomass in 1960, which was 2.3 times 100. And that is going to give us the percentage decrease, how much it's gone down by. And it's gone down by a lot, basically. So for doing 2.3 minus 0.1, you'll get 2.2. That will get you a mark. Then divide them and times by 100 for another mark, change over the original. You'll then get 95.65. Which, yeah, 95.65. But remember that it asked us to round it up to the two whole numbers. So we're looking at 96%. And that's because 6.65 is bigger than zero point 0.5. So you round up rather than down to 95. So hokey dokey. Let's have a little look now. We got the next question. So zinging down, 7.7. Too many herring were caught between 1960 and 1977. Herring can live for up to 12 years and begin to reproduce when about 3 to 4 years old. Laws are being introduced to help conserve herring. That means keep the numbers up. 1977 to 1981, a 4-year law, herring fishing was totally banned in the North Sea. 1984 to the present, the control of the mesh size of the nets. And 1997 to the present, fishing quotas were introduced. And 1998 to the present day, herring fishing was banned in the breeding grounds during the breeding season. You can see these big fat 8-year-old herring get stuck in the net, and the 4-year-old herring can just swim on through. Okie dokie, we have another graph here of the biomass against the year. Evaluate the effect of these laws on the conservation of herring stocks. Now, it does say to us we must use the data from figure 11 and information from figure 12 in our answer. So we need to actually use numbers here. When they ask you to use data, make sure you use numbers. Now, one of the things is the 4-year-old herring can get through the hole in the mesh. Why is that important? Well, small herring haven't had kids yet. By the age of 3 to 4, they haven't had kids. So if you eat them then and kill them then, you are destroying the population size. Whereas if you allow them to have children first and then you kill them and eat them, you're not damaging the population size so much. So it's important that the law was made to allow them through, and this will have an effect of increasing the population size. Okay, there are a bunch of other laws here, and if you look at all the data, so we can see here from 1977 to 1984, the herring went from zero point 0.1 to zero point 0.9, roughly. My lines are absolutely awful. And then from the 1997 law, they went from 1.15 to 1.25. Oh, drawing these lines is just disgusting. And then so forth. So if you look at all the dates and the deadlines, you'll find all the answers. And the law they introduced in 1998 showed an increase from 1.25 to the present. Some of the things, as it's an evaluate question, you need to say some things against these laws. It's hard to tell which law is having the impact because all the laws overlap each other. So, like, we had a law start in 97 and we had a law start in 98. So, which one of those is having the effect? All we can see is that there's a correlation with the laws being introduced and the increase in the population of herring. We can't say it's a causal effect. We can't say specifically it's this law that is causing the herring population to increase. There could Okay, question 8. Sickle cell anemia is an inherited condition that affects red blood cells. Sickle cell anemia is caused by the mutation in the gene for hemoglobin. Hemoglobin is that red pigment found in red blood cells. A person who is homozygous for the normal hemoglobin, HaHa, produces normal red blood cells. A person who is homozygous for the mutated allele HS produces red blood cells with abnormal hemoglobin, has red blood cells that can form the altered shape, and has sickle cell anemia and becomes ill. A person who is heterozygous has both normal and abnormal hemoglobin in the red blood cells and has something called sickle cell trait. They're generally healthy, but they can become ill in certain particular circumstances. So, question 1, 8.1, that is, give the reason why a mutation in the gene coding for hemoglobin could be harmful. What's the job of hemoglobin? Do you remember from Unit 2 and your paper 1 exam? Hemoglobin is a pigment that picks up and holds oxygen in the red blood cells. If you have a mutation in the gene coding for hemoglobin, its shape may be changed and it may not be able to carry as much oxygen. So we need to be able to say reduced amount of oxygen transport. So if hemoglobin has the wrong shape, it will not be able to carry as much oxygen. And that's our answer. And so symptoms of this might be fatal. You could die because you don't have enough oxygen, which means you can't get energy from respiration, or it could just leave you incredibly fatigued, not able to exercise much at all, as you just can't carry that oxygen to the muscles to do respiration and contract those muscle tissues. All right, let's move on. Okay, 8.2. Figure 13 shows some red blood cells from the blood of a person with sickle cell trait. Calculate the proportion of cells in figure 13 that have an altered shape. So let's circle all the ones that look a bit weird and don't look like the donut shape. We've got 4 kind of sickle half moon shape. But what's the total number of red blood cells there? So now we need to count up the total number of red blood cells and then work out the proportion of the total that have the altered shape. So if we now count up the total number of red blood cells, we will see that we have, if you count each of these, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You also, when you're counting the total, obviously include all of the cells, include the 4 altered ones. And it comes to something like 17. We have 17 total cells. So the ratio is 4 to 7. You could simplify that as a ratio, or what you could do is just do 4 divided by 17 and you'll get the proportion. You could turn that into a percentage by doing 4 divided by 17 times 100. It's totally up to you. Don't forget to hit the like and subscribe to the channel. Share this with your friends so everybody can get a chance to do really, really well in their GCSEs. Thanks for that, guys. Hit me up in the comments if you have any specific questions or walkthroughs you'd like me to do of particular papers. I'll basically work as hard as I can to get you the stuff to help you pass and get the best grades possible for you. Okay, figure 14 shows the inheritance of some genes in a family. This is called a pedigree diagram. And you can see the white square is an unaffected male, white circle, unaffected female, black is male with sickle cell anemia. They must be big HS, big HS, homozygous for the sickle cell. Female is a black circle too. A grey square is a male with the sickle cell trait. That means they're HAHS, and a female is a grey circle with the sickle cell trait. Now it says person 8 and 9 in figure 14 are expecting a second child. Determine the probability the child will be a girl, and what's the probability they will have sickle cell trait. Draw a Punnett square diagram, identify the phenotype of each genotype, use the symbols HA and HS. So let's zoom in on 8 and 9. So male is unaffected. They must be HAHA, and that must be their genotype because they're unaffected. And we were told in the STEM that HAHA is someone who has normal hemoglobin. Now, 9 has got the trait, so they have HAHS. If they had HSHS, the woman would be a black circle, but she's a grey circle because she just has the trait. I draw a Punnett square like this, noughts and crosses board, scribble out the first one, write male to identify the male. We've got HAHA. Now female, I can only fit an F on here, and we're going to do HAHS. And now determine the genotype, the combination of genes for the offspring. So the first one is HAHA. They are unaffected. Okay, so they're normal. I could write the word normal or unaffected. They do not have the sickle cell or the sickle cell trait. Now the second one is HAHA. They are also unaffected. Now we have the third probability or possibility of a child. There would be HAHS. Now they would have sickle cell trait. They would have it be grey, coloured in grey. And the fourth one is HAHS2, and they would have the sickle cell trait as well. They would not have full-blown sickle cell anemia. Now, we were told what's the probability of them getting sickle cell and what's the probability of them being a girl. Well, it's 2 out of 4 that they get the trait. So that's 0.5 as a probability, okay, or a 50% chance that they get the trait. But we were also asked, what's the chance of them being a girl? And that's 0.52, right? That's 50%. You remember, I'm doing a Punnett square here. If we do the XY chromosome from sperm and the XX from eggs, and we cross those over, it's a 50-50 that you'll get XX. So we need to do 50% times 50% or 0.5 times 0.5, and the answer is 0.25, and that will get you 6 big fat marks or 25%. Okay, so the chance of you having a girl with the sickle cell trait is 25%, or a probability of 0.25, or 1 in 4, okay, or 1 to 3. Let's go. Okay, final part of question 8.4. Without medical treatment, people with sickle cell anemia are frequently ill and have a reduced life expectancy. The malarial parasite cannot live in the red blood cells of a person who has the HS allele. A scientist stated, it is an advantage for people to have the HS allele in countries where a malaria occurs. Evaluate the scientist's statement. So when you see the word evaluate, you really want to come up with at least 2 things for and 2 against, and you need a final judgment. You have to give your opinion with evaluate and back it with a bit of evidence. Okay. Now, as it's 3 marks, you could do 2 for, 1 against, or you could do 1 for, 2 against. Minimum of 3 points and then an opinion. Okay, let's check it out. If you have HAHS, you won't get malaria. You're protected from malaria. So the scientist is right. You can't get the sickle cell anemia. However, if 2 parents have HAHS, they could have an offspring. One of their children could get sickle cell because you could get the HSHS coming together. And that's obviously not good because they're going to get severely ill. And some people may even get ill. Remember, it said under rare circumstances with HAHS. So even the parents could get ill in certain circumstances having the HS. However, malaria can be fatal. So I would say, personally, it's better to have the HAHS and be protected from the malaria. If parents are HAHS and HAHA, then some offspring will have protection against malaria. We could also say if parents are HAHS and HAHS, then 50% of them will have the protection against malaria, won't they? Yeah. Or if in the example I've given above there. Overall, I disagree because some of the offspring could get sickle cell if both parents are HA/HS. So I'd say the scientist has made a statement that it's just generally good for people to have HS. Well, it's not good if both parents have HS, in my opinion, because there's a chance that a child will, there's like a 1 in 4 that the child will have sickle cell anemia, which is really bad. Okay, now we're on to the final question 9. Figure 15 shows two modern species of Galapagos finch and their classification. We've got medium ground finch and the small ground finch. And down the side, it's got kingdom, class, and genus. So we are missing some of the groups or the levels in the hierarchical system of classification that the older Carl Linnaeus or whatever his name is came up with. And the way to remember the order is king prawn curry or fat greasy sausages. There are some other ones like king's play cards on fat girls' stomachs. There's all sorts of different ones. So you just have to remember that the king prawn curry, well, the P stands for phylum, or stands for order, fat stands for family, greasy is genus, and sausages is species. And that's how I remember the order, and that will get you 2 marks. Let's zing on. So let's go to 9.2. Give the binomial name of the medium ground finch. Use the information in figure 15. Now, the binomial name is always written in italics, and the binomial name is the genus followed by the species. So the first word in the name is the genus, and the second word is the species. So they're right at the bottom here, the genus and species, right at the bottom of the classification hierarchical system, and it's Geospiza fortis. So Geospiza is the genus and fortis is the species. The binomial name, bi meaning two name system, saves you having to name the whole thing and call it Animalia, Chordata, Aves, whatever. You just can name the last 2 parts of that system. So in each of the species of finch, there is a variation in the depth of the beak, shown in figure 16. Figure 17 shows the relationship between the beak depth of a parent bird and the beak depth of their children, which is the offspring. So we can see there's a correlation. As the parent's beak depth gets bigger in millimeters, so does the offspring. Give evidence from figure 17 that beak depth is an inherited characteristic. Well, exactly that. We can see that the birds with bigger beaks have offspring with bigger beaks, essentially. So as the bird parent beak length, beak depth, I keep saying length, but as the bird parent beak depth increases, so does the mean beak depth of the offspring. So that's the evidence that it is an inherited characteristic. 9.4. Scientists suggested that more than one gene controls the beak depth. Give evidence from figure 17 to support this. So, how do we know that the genes are polymorphic, which means this characteristic is polymorphic? It has more than one gene that controls the beak depth. And the idea here is that there is lots of variation in beak depth. So even if you look at a particular beak length in a parent, there is lots of variation. So all different types of beak depth. Now let's get on with the next question. Figure 18 is a map of the Galapagos Islands. We have one called Daphne, one Isabella, and Crossman. Isabella Island, the medium ground finch and the small ground finch are found, but on Daphne only the medium ground finch and Crossman only the small. And we can see here, this shows the percentage of the population and their beak depth. So on Isabella Island, it looks like the small ground finch has a certain size of beak depth, and the medium ground finch has a different size of beak depth. The medium ground finch and the small ground finch both feed on seeds. The size of the seeds eaten by each bird depends on the depth of the bird's beak. The range of the beak depth of the medium ground finches on Isabella Island is different than on Daphne Island. What might have caused the difference? So, why is the same bird living on 2 islands, but one set of birds has a longer beak than the other? Well, the birds arriving on Isabella Island have a range of beak depth, some large, some small. The variation in beak depth is really caused by the fact that the birds have different alleles for beak depth, i.e., some might be for long beaks and some might be for small beaks. So this variation in beak depth is being caused by different alleles. Where do we get different alleles from? They're caused by mutations in genes. That's how new alleles arise. There must be competition for seeds on Isabella Island, which means that the birds with the larger beak depth or the bigger beak depth have got an advantage and can get to the seeds more easily. Therefore, they have a bigger food supply, more energy, more likely to survive and pass that beneficial allele on to their offspring. And then over many generations, we end up seeing that the birds on that island all have large or a a high beak depth. God, I couldn't get the words out. Okay, on to the final part of this whole paper. So let's take a little look. 9.6, figure 19 shows the two species of finch live on Isabella Island. Only one of the species lives on Daphne Island and only one on Crossman Island. So Daphne Island only has one type of finch. Isabella has both types of finch, suggests why both types of the bird are able to live together on one island. And the idea here is that the beak depths of the two species are quite different on Isabella Island. So one of them has a very high beak depth and the other one a short beak depth. We can assume this means they avoid competition between each other as they eat different types of seeds or different size seeds. And so, therefore, they're not competing with each other, which makes them more likely to survive by avoiding competition. And that's it done for this exam, guys. Well done. Check out the other walkthroughs for the other exams. Good luck with your exams. Don't forget, let's get to the marks.
You're still here? It's over.